Okay, so now we'll continue on with the introduction to archaeogenetics. And this will be given to you by Selina Carlhoff and James Fellows Yates, both of whom are doctoral researchers in the Department of Archaeogenetics. Selina has a background in marine biology, and she completed a master's degree in it at the University of Southampton. And she's interested in the population genetics of Southeast Asia, and her research focuses on the transitional periods in the history of Southeast Asia and how these affected the genetic structure of the people living there. And James is a biomolecular archaeologist and bioinformatician, and his current research focuses mostly on ancient DNA content of dental calculus. And today, both of them together will present the introduction to archaeogenetics, focusing on how genetics is important to reconstruct human history. So you can take it away.
Yes, thank you so much, Carly. And also welcome from our side to this second introductory lecture of the day. And yeah, myself and James will give you an overview of what archaeogenetics are and the topics that we'll be covering. And first are some definitions and also the history of archaeogenetic research, and then why ancient DNA research is so challenging, and where we'll also go into some of the laboratory methodologies that we need to extract DNA from ancient material. And in the second half of the lecture, we'll introduce you to some examples where ancient DNA work has added substantial information to the field, and we'll also focus on one example specifically where archaeogenetics, archaeology, and linguistics really worked all together to support a theory. So without further ado, we'll just get right into it. So what is ancient DNA or archaeogenetics, and what is its history as a field? So archaeogenetics as the word is just made out of two compounds. It's a compound made out of two words. First is archaios, which is Greek for ancient, and genetics, which is the study of heredity. So plainly, archaeogenetics is simply the study of ancient genetic material, which is also known as ancient DNA or aDNA. And as most of you know, DNA contains the genetic instructions for the development, functioning, growth, and reproduction of all known organisms, and also many viruses that live on this planet. So in archaeogenetics, we combine the knowledge of ancient and present-day genomics to understand the evolution and development of species in a large variety of contexts. And another synonym for archaeogenetics is also paleogenetics or genomics. You will see variations of these words if you look for publications in this research field. So, and since genetics and the development of the genetic structure is only really achieved in the 20th century, also archaeogenetics is, as a child research field of genetics, is also still quite young compared to especially archaeology and also linguistics. And for the longest time, it wasn't even known that, or thought possible, that the very small molecules within cells survive after the animal has died. So it was not until the 1970s where several researchers actually could show that proteins and organelles in cells would survive in material which was older than 100 million years, or insect tissue which was preserved in amber. And you can see in these two pictures one polyacrylamide gel where they characterized these proteins from the mollusk shells, And in the lower picture, you can see where they found these ribosomes, which are also little organelles in the cells in 40 million year old insect tissue. So that was quite exciting, of course, but ancient DNA studies remained really quite challenging. So it was not until 1984 that the first DNA sequence from an extinct species was extracted and could actually be used to place the quagga, which went extinct in around 1883, but is preserved in museum specimens. And the quagga could be placed within the horse family, and yeah, you can see a quagga in the picture on the right. And in 1985, Swante Paavo, which you might have heard about, was actually able to clone human ancient DNA from an Egyptian mummy. And this showed that far older DNA may be preserved in still reproducible form, and that you could extract both nuclear as well as mitochondrial DNA, even after millennia. And even though these publications were very exciting for the field, there were also a lot of controversies, especially about the Egyptian mummy DNA. And around this time, there was also one of the key technological improvements that really sped up the development of the field of archaeogenetics. And this was the development of the polymerase chain reaction in 1983. And with this technology, they were able to really rapidly and quickly reproduce the DNA that they would find in ancient tissue. And this enabled extraction of ancient DNA from ancient human brain tissue, the dried skins of extinct species from Australia or New Zealand, and also from the fossilized remains of plants and insects. But so far, the extraction of ancient DNA was always limited to skin tissue, which, as you can imagine, is not very well preserved in the archaeological record, since skin obviously decays quite quickly. So when in 1989, two different teams of researchers were able to show that you could extract ancient DNA from bone, it was quite a revolution as well. Because of course, bones are much more available in the archaeological record than skin or other organ tissue. So with bones being available to archaeogenetic studies, many researchers tried their best to investigate archaeogenetics and, for example, posted the first studies looking at prehistoric Pacific Islanders and also prehistoric North American populations. But all these studies popping up about human DNA quickly became quite contentious. And even older studies were examined under a more critical eye. And also the ancient mummy study, which was one of the first ones in 1985, was actually shown to mostly contain modern DNA. So no ancient DNA actually. So and actually contamination became the biggest issue and the biggest controversy within the field. So when Klingstedt et al. In 1994 were trying to extract the first Neanderthal DNA sequence, they took extreme safeguards to avoid contamination in their samples, where they repeated all experiments in 2 different laboratories and had numerous controls just to control for the background contamination that might be present in the laboratories. And in 2000, this all culminated in a publication by Cooper and Poyner, which actually claimed that if you do ancient DNA, you have to do it right or not at all. And in this publication, they suggest some criteria of authenticity to control for contamination and ensure reproducibility. So another technological step then was in 2005, when next generation sequencing made genomic sequences really widely available. It was much faster than older sequencing techniques and also cheaper to produce many, many samples, sequences. So you can see here in this graph I compiled from the PubMed database. You can really see that after 2005, and particularly after 2015, the number of publications in the fields paleogenetics or archaeogenetics or dealing with ancient DNA has really, really just doubled in just a very short period of time. And of course, the bar for 2021 is still in progress. So I'm sure it'll be another peak probably. But yeah, the field is still has a history, but it's still relatively new, and there's new technology coming out all the time. So it's really quite exciting.
But going back to sort of what Selina said about the history of ancient DNA, it's not easy, and there's a variety of reasons why that is. But to understand this, we have to go right back to the fundamentals and look at the structure of DNA. So in all of our cells, on our human body, for example, all of our DNA is wrapped up in chromosomes, which are very, 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 very tightly, super tightly coiled long strands of DNA. But when you unravel this, you get the form of this double helix, which I'm sure most people should be familiar with, which is made up of two strands of molecules. And these molecules are made up with these equivalent of building blocks, as you can imagine them to be. And each building block is made up of 4 possible nucleotides, which are these little A, C, Gs, and Ts that you can see here, or nucleobases as described there. And each strand of these nucleotides are kept together by something called a sugar phosphate backbone. And the important thing to remember about these different nucleotides, the C, G, A, and Ts, is that they form complementary base pairs. And what we mean by this is that every time you find a T, this will always be bound with a, sorry, every time you find an A, this will be bound with a T. And anytime you find a G, you will find a C, and vice versa, because of the different number of hydrogen bonds you can see between the two here. And this is very important for ancient DNA. And the other important thing to remember is to make new DNA, you have to go through a process called replication. And this has been exploited extensively in molecular biology. And so a lot of our lab techniques are based on this basic concept, which is to make a new strand of DNA, all you have to do is separate the two strands from each other. And then you get enzymes such as polymerases, which basically run along an exposed strand and looks what is on that strand at each position on the nucleotide sequence. So if you find an A, then it will find a T, and pull a floating around T nucleotide out of the soup and stick it onto the new strand. And then it goes to the next one. If it's a G, it'll look for a C and pull that in until it's bound. And that way you can see you get these two new strands of DNA, which is all based on this existing one here. But the important thing about ancient DNA is that it's degraded. It's very broken down, it's damaged, very much like a pot in archaeology. You throw it in the ground, over time it will get squished into lots of fragments, and then the edges of those fragments will get damaged. And this rate of degradation is extremely variable. So in stable, cooler environments, this seems to facilitate DNA survival, which is why you'll see a lot of ancient DNA studies are being performed in the Northern Hemisphere in particular, but also in places like Siberia, where it's very cold permafrost environments, and you get nice preserved whole mammoth bodies. And in contrast, when you use a very, very warm or wet environment, the DNA does not seem to survive very well. Although admittedly, this always depends on the microenvironment of every site. So if you find a nice cool cave in Africa, for example, which is typically very warm and not so good for ancient DNA preservation, the cave will actually preserve the DNA a bit better than you would expect. And this degradation happens through a variety of biological and chemical processes. So the degradation can happen primarily because once the body dies, there's no more enzymatic repair, which is occurring while you're living. And so there's no enzymes or anything going around fixing all the mistakes that keep happening. But once you die, also, there are lots of things which start basically chomping away at the DNA. So for example, there's a suite of enzymes called nucleases, and these will naturally break down DNA when they're normally used actually to sort of maintain the quality of DNA in your body. But these basically go wild and start chomping everything up it can find. But also microbes that are either in your body or in the soil will also start degrading the DNA, and that will also make them fragment more and more and more and more. And it basically makes these very long strings of DNA into these very small fragments. And this is very important when it comes to ancient DNA, because there are two main types of ancient DNA degradation characteristics we have to tackle with when we're analyzing the DNA. So as you can see here on the left, in the upper plot, we have a given nucleotide and then a series of strands, two strands. So the P indicates the phosphate backbone on both sides. And what you can see on the lower two plots is that actually this is broken. In the very bottom nucleotide on the left, the phosphate backbone breaks. And this means the strand is not being held together anymore. And these hydrogen bonds are actually quite weak. So it's very easy for these nucleotides on one strand to basically float off and go away into the ether, basically. And what you're left with is basically a strand break. So it converts one long molecule into two shorter molecules. But it also exposes one of the strands, the nucleus of one of the strands at the end of the molecule. And when you do this, you're exposing a nucleotide, on the exposed strand, you're exposing this nucleotide to other chemical processes. And typically this is hydrolysis, and cytosines are particularly susceptible to hydrolysis. And what this means is that the amine group here, the C, gets damaged or modified, and it converts into something called uracil. And now uracil is actually not used in DNA, but rather RNA, which is a different type of nucleic acid. But when we go into the lab and we start to replicate the DNA, to what we need to do during the laboratory methods, is that our polymerases misread the uracil, and basically will find, so rather than getting a G, it'll get an A. And then when you replicate it again, as you're only supplying the DNA nucleotides, the complement of the A will be a T. And what this means is that actually when we read the DNA, even though it was originally a C, we get a T. And importantly, these conversions, these miscoding lesions, as they're called, from C to T, are only happening on these exposed strands at the end of molecules. And so when you actually look along the molecules, what you see is an increase in frequency of these miscoding lesions at the end of molecules. So here on one strand, the 5' strand, you see the red line, which corresponds to the C to T transitions, or lesions, I should say, is much more elevated right at the beginning of the DNA molecule. And conversely, on the other strand, because of this complementary base pairing, we see this increase in G to A. And while this is annoying, because it means that you don't actually have the original sequence from the cell of the dead organism, it does give us an indication that this is true ancient DNA, because it's undergone these degradation patterns. And so because of these very short molecules and also this damage, this makes ancient DNA difficult, but equally, on the other hand, it allows us to authenticate our ancient DNA to know it's real. And the reason why that is important is because of contamination. Whoops, I went too far there. As our DNA is degrading so rapidly over time, it's getting smaller and smaller and smaller and smaller. Some of it basically just disappears into the ether because it's too small to recover. We have very, very small amounts of the original DNA that was in our sample. And at the same time, over time, we're getting a variety of new modern contaminating DNA from the environment. So this can be in the form of microbes, this can be in the form of animals. So for example, famously, there was a study where they measured sheep peeing on a plot of land and measuring the level of sheep DNA that gets into the ancient samples. Also, once you've excavated, for example, a bone, the excavator will be handling the bone. So human DNA will be going onto the bone. Then someone will lick it, for example. So that's a common method in excavations to detect whether something is a bone or a tooth. And then when you go to the museum, this will be sitting in another environment, and all of these places are bringing this modern, very long, high-quality DNA into the samples, which makes it very much that a lot of the challenges we have with ancient DNA, on top of this fragmentation and the damage, is that we have to really hunt for the needle in the haystack to find the DNA we want to analyze.
Yeah, so there's a number of laboratory methods where we try to exactly do this. Find the little piece of ancient DNA that is still left in the bones. And sometimes the percentages of so-called endogenous DNA, so the DNA that really belong to the organism that we are looking at, can be below 0.1%. So it's really quite difficult. Before we go, of course, into the lab, we have to find and think about the potential sources where we could extract or try to extract ancient DNA from. And I've listed the most common ones here, and particularly the petrous part of the temporal bone. For studies of human genetic history, this is really quite the most popular one, and the part of the body where it's been shown that most DNA is preserved. And this little bone that's otherwise quite unknown also houses the inner ear, and because of that, it's very, very dense. And there's also not a lot of circulation, so blood circulation within this bone. And since it's also within the skull, the petrous bone also seems to be quite protected against other forces that destroy bones during their degradation. So, and another really quite hard part of the human body and the human skeleton are, of course, teeth. And for teeth, because the dentin is, I think, one of the hardest materials generally, there's also been many successful studies extracting DNA from within the crown chamber of teeth. And of course, also regular other skeletal bones are also sources of ancient DNA. But these all, of course, tackle like humans, and this also so do skin and hair samples. Whereas with ancient DNA, you can also study, of course, animals, for example, through the analysis of parchment. You can study plants through archaeobotanical remains, and you can also investigate what was in a ceramic vessel with ancient DNA. So once we've selected a suitable sample for ancient DNA study, these samples usually come with us into so-called clean rooms. And these are dedicated spaces where usually only ancient DNA work is done, just to reduce the potential contamination that James talked about. And in clean rooms also have other features such as positive air pressure, and most of them are also really strictly cleaned, for example, with UV lights. And within clean rooms, of course, if we're taking such good care to keep the room clean, we also have to make sure that we don't bring anything into this very closed off environment. So protective lab gear is definitely on the agenda. And in the picture, you can see a very good example of what people who work in the clean room look like. There's, of course, face masks and these full body suits. The dedicated boots that you only use in the clean room, and we actually even the clothes that you wear under the suit are usually only worn in the lab. So you don't take any just everyday street clothes. It's just all to make sure that you don't bring anything from the outside into the clean room. And the most important part are, of course, also the gloves, where you wear several layers that get, and they also get changed after everything you touch. If it's another sample or another tool, there's a lot of glove changing, believe me. So it also just takes some time to get dressed to go to a clean room. It's quite a commitment. And another big time commitment within the clean room is the decontamination of all used materials and the workstations and all tools with UV light and bleach to destroy even the last bit of DNA that might be left on the surface where you just handled a sample. And the more, I guess, so these are all protective measures that we try to do to prevent contamination. But we still have to, of course, check if something did slip through the cracks. And for that reason, we use blanks, which are samples which are only filled with water. So these little pure water samples just get taken along through the entire process, and if are also sequenced in the end. So you can see if there's any DNA in these blanks where there's not supposed to be any, then you likely had a problem during the experiment and have to be careful with the interpretation of all your other data that you produced in the same run. So it's really quite pivotal to studies to check your blanks. And on the other hand, we also want to control for human error. So we take positive controls along, which are samples that we know work really well, to know that if in the end none of the, or some of the experiments didn't give any results, then if the positive controls also didn't work, that usually means that you forgot to put some chemical into a reaction. And so it's easier to troubleshoot where the intrusion into where the problem happened. But luckily, this doesn't happen very often, neither that the blanks are contaminated, nor that the positive controls does not work out. But it's very important to keep along and to check also for background contamination that might be present in the laboratory. So if we get to the actual preparation of the bone or the tooth, here these are the two examples that I picked because they are the most common. Just to give you short insight into the process in the lab, with petrous bones, we usually cut them open in the middle with a little circular drill, and then they look, you can see in the second picture, kind of a schematic of what they generally look like. And in this red marked area, which is exactly around the channels where the inner ear is, this is the very densest part, and this is where we use these dental drills to drill for the bone powder that we use for extraction. Whereas for teeth, we also cut the root off and then drill within the pulp chamber and also generate bone powder this way. Of course, you wouldn't mix these. This is just for the schematic. The bone powder is then stored at -20 degrees until you're ready for extraction. And after, of course, each sample, you have to clean everything in the workstation. just to make sure that there's also no cross-contamination between the samples. So it's quite likely that it takes longer to clean in between each sample than actually drilling, but you just have to make sure. So if we come to DNA extraction and we have the bone powder, here we use mainly use a protein called proteinase K, which is part of the extraction buffer that we mix, which breaks down the cells of the bone that are still, of course, intact in the bone powder, and it really just breaks the membranes open to release the DNA and everything else that's in the cells into solution. And to do this, it's incubated for around 24 hours and then centrifuge. So you separate the still solid portion where there might still be some bone powder still or other organic material that wasn't broken down from the supernatant, which is what you call the liquid that separates that flows on top of the solid portion. And in the supernatant, you should have all the DNA and also lots of other chemicals and molecules which are put into solution. So to separate the DNA really from everything else that's still in solution, we use the binding buffer, which contains guanidinium and an alcohol, and use these in combination with silica columns where the DNA binds into the walls, and then we wash through the column to really remove everything else. And then in a final step, we use a chemical called TET to release the DNA from the walls of the column. So you should now have the DNA suspended in TET, and you can discard the column and store the liquid with the DNA again at -20 degrees.
But before we can actually start working out the sequence of nucleotides in our DNA, we do this thing called next generation sequencing, as Selina mentioned earlier in the history of ancient DNA studies. And this is a way of sequencing millions and millions and millions and millions, or even billions nowadays, of DNA molecules at once. But before we actually send it to the sequencer, we have to make some modifications to our DNA molecules before we can do so. And so we do this in something called a library preparation. And what library preparation does is firstly repairs the end of the molecules. So you have, you remove the single-stranded overhangs, which may occur either by clipping them off at one end and filling them in at the other end. And this is in a process called blunt end repair, as you can see with the polymerase there. And this is where this C to T miscoding lesions typically come in. Then we have to add things called adapters. Now, to be able to image the molecule, to know what sequence of nucleotides there are on it, you need to make sure we can immobilize the DNA molecule somewhere to basically start recording these nucleotide sequences. And to do this, what we do is add these things called sequencing adapters to the end of the molecules. And these are synthetic DNA molecules where we know what the sequence are. And so by getting the complement of those, we can attach these in the machine and allow us to anchor our DNA to the machine to do this imaging. But in addition to the adapters themselves, we include something called indices. And now what these indices are basically equivalent to a barcode in the supermarket. So they are a unique sequence of nucleotides, also synthetic in this case, which we assign to each sample that we want to sequence. And what this means is that if we sequence these barcodes along with DNA molecules themselves, we can, after sequencing, separate each DNA molecule to be associated with just the sample that you added. And what that means is then we can sequence loads and loads and loads of samples all at once on one machine. And now, typically, up until about 2 or 3 years ago, this whole process of library preparation would take about 3 days. But nowadays, with improvements in molecular biology, there are automated liquid handling workstations, aka robots, which allow us to do this in as few as 9 hours to convert our DNA molecules extracted in the previous step to being ready for sequencing on a sequencing machine. And I'm going to actually cover both the light preparation and also sequencing in a bit more detail tomorrow in the Intro to NGS processing workshop. But what this basically consists of is, like I said before, immobilizing our DNA molecules using the adapters to a platform, and then basically start adding the new nucleotides based on a single-stranded molecule. So using the same process, the replication, as I mentioned earlier, and each time you add a new molecule, you record what new nucleotide has been added. And this all happens on these machines, such as this Illumina HiSeq 4000, I think it is. Yeah. Good.
Great. So after sequencing, we of course have the genetic sequences stored on some really big computing cluster because they take a lot of storage space, and we can then use them to investigate the kind of problems or research questions that we might have. And in the next section, we want to introduce you to some popular examples where ancient DNA has been used. And to get started, the first really big section where ancient DNA is being used today are population genetics. And just as a quick introduction, since Jena and myself will talk about population genetics more in detail also tomorrow, but in the afternoon, just a quick definition, so you know what I'm talking about at all, is that population genetic studies look at the genetic differences within and also across populations and try to explain the dynamics at how this population evolved. And the processes that we're looking into to find that out are, for example, natural selection, genetic drift, mutations, and also, of course, gene flow and admixture processes between different groups. So with ancient DNA, it's possible to directly compare the genetic variation at different time points, and from that, try to infer what happened, what demographic processes happened to result in the genetic variation we see in later time periods. And some examples of where population genetics using ancient DNA were really quite key in establishing the relationship between different groups were, of course, looking at extinct hominins, such as the Neanderthals. And the first remains that were called the Neanderthals because they were discovered in the Neanderthal, which is a valley in Western Germany. They were discovered in the mid-19th century. But later finds showed that Neanderthals occupied vast parts of Eurasia between around 400,000 and 30,000 years ago. And even through early morphological comparisons, they theorized that Neanderthals were either direct ancestors of anatomically modern humans, so us today, that they contributed genetically to anatomically modern humans, or that modern humans completely replaced Neanderthals, and there was no real genetic contact between the two hominins. And some early mitochondrial DNA studies indeed showed that all Neanderthal mitochondrial DNAs fell outside of the human variation, which suggested that there was no genetic contact between these two hominins. However, later studies using nuclear DNA showed that indeed there is a quite small but significant proportion of between 1.5 and 2.5% of our genetic ancestry that everyone outside of Africa can trace back to the Neanderthal, actually. And more advanced studies have shown that there's also indications of selection on these genetic variants that we can trace back to the Neanderthals. And the other extant hominin that has received quite a lot of press coverage as well are the Denisovans. And they are actually only known from some really small individual bones recovered from Denisova cave in Siberia, which also gave the name to the hominin, and also Tibet. And they all date to around 160,000 years. And from the genetic, they seem to share a common origin with Neanderthals. So they are more closely related to the Neanderthals than to modern humans. And even though the archaeological remains have only been found in this quite separate in a very small part of the world, it seems that the Denisovans contributed to quite a vast group of modern humans as well, similar to the Neanderthal. But here the contribution is quite regionally differentiated. For example, the highest amounts of around 3% are found in present-day Near-Oceanians on Papua New Guinea and in Australia. But there's also traces of Denisovan ancestry in East and South Asians and also in Native Americans. So there's really quite large regional differences, which seems to suggest that instead of one admixture event, which seems to have taken place for the Neanderthals, for the Denisovans, it's likely that there were several introgression events and of differently related Denisovan groups. And there's even the evidence for distantly related Denisovan ancestry, so from two different Denisovan populations, can both be found in Papuans today. But there's still a lot to learn about the Denisovans, and there's new publications coming out about the Denisovan every year. So that's really, really quite an exciting part of not directly human, but like human-related history. But if we move further into the more close, the not so long ago past, one of the big studies that had really had a huge also media impact was the study by Wolfgang Hack et al. In 2015, investigating the Steppe expansion. And Wolfgang will also be there in the roundtable discussion on, so be sure to look out for him there as well. But in this study in 2015, they sequenced 69 Europeans dating between 8,000 and 3,000 years. And really mostly from Western Eurasia, but through all different time periods, from hunter-gatherers really far in the past, all the way through the Neolithic, so when people started to use agriculture, into the Iron Age, which was only around 1,000 years BC. And in this study, they found that when you model, so this is an admixture proportion graph, and I'm just quickly going to walk through it because you will encounter these a few more times and maybe you're not that familiar with them. So in admixture proportion graphs, you usually see two or more, the result of modeling of two or more admixture sources for one specific population. So for example, at the bottom here for Starchevo early Neolithic, you can see that the whole bar, the whole 100% of the ancestry can be modeled as early Neolithic farmers, which are here displayed in orange. And if you go through time, if you go further into the future from that point of time, you can see that there's a blue component coming in, which is representative of hunter-gatherers who inhabited the region previous to the arrival of the farmers. But then around in the late Neolithic, you can clearly see that there is a large new component coming in, which is represented in green, and that is related here modeled by the Yamnaya culture, which is from the Eurasian steppe, so quite far away from all these Western Eurasian sites, but clearly present in all of the younger individuals from this study. And actually, this Yamnaya-related ancestry is present today in all European populations in varying proportions, but it's there undeniably, with the largest proportion actually in Norwegian people from today. However, the steppe expansion did not only happen towards Western Eurasia. Later studies also showed steppe influence in Eastern Eurasia, or Central Eurasia rather, even all the way to North India. So there seems to have been a contemporaneous expansion into the West and the East from the Central Eurasian steppe. And this expansion has been hypothesized to be related to the spread of Indo-European languages as well. So it really massively impacted the Bronze Age of all of Eurasia, or the majority of Eurasia, and really can be traced into the present for also many groups.
It's not just humans that we can directly study. We can also study other animals. So if there are any zooarchaeologists attending who are also interested in using genetics to understand maybe human and animal interactions, there's quite a few examples out there of these sort of studies. So for example, in 2016, Gloria Fortes et al. Were analyzing different cave bear materials in caves across northern Iberia. And when they were looking at the mitochondrial DNA, so this is the DNA inherited maternally, they stumbled across an interesting pattern where when they were looking at change over time in one cave, that in the cave bears, so these are extinct species of bears, the same sort of haplogroups were preserved, so the same throughout time. Whereas in contrast, there was much more diversity with the brown bears. So there were different lineages of brown bears living in caves at different points of time. And what this suggested to the authors is that over thousands and thousands of years, cave bears kept going back to the same caves repeatedly. It was almost like a homeland or hometown. Whereas the brown bears didn't care so much and would just go wherever they sort of found was comfortable. And what the authors interpret this as is that it was a possible indicator that cave bears had quite predictable behavior, making them more at risk or susceptible from human hunting, and that maybe possibly contributed to their eventual extinction. But it's not just macroorganisms we can study. In archaeology, particularly, we're dealing with dead people. But obviously, an obvious question is, why are they dead? And so in archaeology and history, there are a variety of traditional methods of identifying disease. So there's obviously historical records that describe the diseases themselves, but often you can get conflicting accounts depending on who wrote it and how they described them. Archaeologically, we can look at attritional or mortality profiles. So these are looking at the demographics of a particular cemetery or site and see, are they skewed from a typical demographic makeup? So for example, having 25% children, 25% old people, and 50% adults. If you see a much bigger adult population represented in the cemetery, that's just maybe a lot of adults were dying for some reason. More specifically, we can look at the remains of the animals, humans and scales themselves by skeletons, and specifically by looking at modification or bone lesions on the skeletal remains. And this is because infections can often lead to characteristic modifications. For example, in the example here, where you see this porosity and sort of massive overgrowth, but these are also quite unspecific to the actual causes, so the agent that caused that particular infection. Also, bone lesions suggest long-term or chronic disease, so it doesn't necessarily mean that the individual died from that particular pathology. And these questions, what about lethal infectious diseases, where you die immediately, so there's not enough time for skeletal signs to develop? But also, what about infectious disease that don't affect the skeleton at all? And this is where ancient DNA comes in, because we can use it to identify the specific species of microbe that may have caused the specific disease or even killed the individual. Now, you can either take the DNA from the bone lesions on the skeleton for those that actually affect bone, but more typically what microbial archaeologists look at is the tooth pulp. So this is inside the tooth, surrounded by the very hard mineral enamel, which, much like the petrous bone, is very good at preserving DNA because it's so hard. And inside this tooth pulp, during life, you have blood vessels. So whatever is circulating in the blood at the time would also be going through your teeth. And when you die, bits of that blood can remain in a sort of a dried form. And so if you extract the DNA from this dried blood, essentially, you can potentially get the DNA which will be infecting the person death. And so the first thing you can do is identify where does this DNA come from? So is it from particular pathogens? So for example, seeing in this top left-hand graph, you have different columns, different samples, and these big red things are known pathogens, which are making up a large proportion of sort of microbial signal of that particular tooth. And that can tell you, well, this person was infected by this particular thing. When you compare your DNA against large databases of sequenced genomes and saying, most of my DNA aligns to this particular genome, or is most similar to this genome. And if you have sufficient DNA, you can actually reconstruct the entire genome of an ancient strain of that infectious disease. So each colored ring represents a different genome. What you can do is when you compare them all, that you see patterns like this sometimes, where you have these missing regions of the genome. And by looking at around the genome for things that are missing or so present or absent, you can potentially identify virulence factors, so things that actually describe why the microbe was so infectious, or like how it actually causes like the disease or the modification, the illness on your body. Furthermore, when you have a whole genome, you can then compare them to modern strains as well. Typically in the form of phylogenetic trees or phylogenetic networks. And with these, you can ask, do you see any geographic pattern? So are your ancient strains always falling with particular strains that are falling from some region of the world today? But also suggesting like a reservoir effect. So this is the source of the disease that might be coming out repeatedly. Also, by doing this comparison, you can also see, does your genome cluster with modern genomes that could indicate the source, the biological source of the disease? So, for example, with coronavirus, the novel coronavirus at the moment, you can ask, okay, is this particular coronavirus falling with bats or pangolins or whatever the current sort of hypothesis is? And that could also give us evidence to say like where ancient outbreaks may have occurred of particular diseases. And also, when you're comparing these trees and you have a known per year mutation rate, which is relatively consistent, we can use this to also date the evolution of the microbe. When did it actually appear? And when maybe, if you can't do radiocarbon dating, for example, when maybe it was present in a particular population. But you might be asking, why should we care, other than the curiosity of why do people die? That's because obviously infectious disease is still a major problem around the world. Diseases such as TB is still one of the largest unresolved diseases, chronic diseases, which kill millions and millions of people per year. And TB is actually identified in the archaeological record from histological or osteological changes. So it shows it's a very old disease. Likewise goes with the agent of Black Death, which is still actually present today in modern US and Madagascar and sometimes causes quite severe outbreaks. And that these are not new diseases is quite important because you want to ask, how do the bacteria over long periods and how are they adapting to various stages of evolution? So in this summary graphic, you can see there's been many historically recorded plagues and epidemics and so on. And we also know that there are various human interventions that we try to do, like quarantine, for example. That's not a new thing. But we don't really know how these sort of, how effective or how these particular interventions worked. How this may have modified the bacteria if they adapted and they weren't sufficiently killed off. And maybe this also gives us a guidance as to why they are still present today. So to look at a couple of examples, one of the most infamous, I should say, epidemics was the Black Death. So this is in the 14th century, killed up to an estimated 50% of the European population. But what caused it was still an open question. Historical records generally pointed towards the bacterium called Yersinia pestis. However, this was still controversial because the behavior or the epidemiological patterns described in the historical records didn't seem to match what we know about the behavior of modern strains. And now early ancient DNA work also indicated this, but this was still controversial because it was Yersinia pestis specialists analyzing these ancient bones while working in the Yersinia pestis lab. And so, as Selina was described earlier, this was not happening in this ultra clean room where you have to change your gloves every 3 minutes, as we do so today. So people were not convinced that this was basically evidence was not some form of contamination. So in 2011, teams in Canada and Germany joined together to analyze the skeletons of using the cutting edge techniques at the time. Exactly, Roberta, which was this NGS sequencing, which I'll be talking about tomorrow. And in this study, they confirmed the presence of Yersinia pestis in skeletons, in this case, taken from the clean lab, not in a Yersinia pestis lab, and from a historically recorded Black Death cemetery in London. So they were able to confirm that that was the agent of Black Death. But in addition, they were able to reconstruct large portions of the genome, which allowed comparison with other strains. And what interesting they found is that the Black Death, so this thing that killed off 50% of the population of Europe in the 14th century, is actually the ancestor of all known outbreaks today, essentially. And this is a big epidemic which is still happening around the world today. So it didn't entirely die off. Now, you may also still be asking, okay, cool, but this is just confirming historical records. Why do we need ancient DNA here? It's already written down. But ancient DNA can also uncover completely unknown epi or even pandemics that exist in the past. So, for example, in 2015, researchers in Copenhagen looked at all the non-human reads of some of the ancient population genetic studies that they were performing there from the Bronze Age. And in this time period, there wasn't really any clear evidence of anything going on there in terms of infectious disease. And unexpectedly, in these non-human molecules, they found reads from Yersinia pestis, which they were like, what? Weren't expecting that. And also this was spread across the whole of Eurasia, showing that this was everywhere already. And when they analyzed the genomes, interestingly, what they found, there were certain genes missing related to transmission of the bacteria as we know in modern day. So Yersinia pestis is carried in fleas, and the fleas are carried by rats. But the changes in the genomes meant that the fleas couldn't properly transmit the bacteria so well, suggesting that actually this early strain of Yersinia pestis worked quite differently. On top of this, in a second paper, this was expanded upon, they found that all of the individuals who were carrying this early form of Yersinia pestis were also the people who had this large Yamnaya or Steppe component of their ancestry. So the individuals who did not have this didn't seem to have Yersinia pestis. And this is obviously very important because these individuals underwent this massive and rapid migration, which Selina alluded to earlier, from the steppe into Europe. And so this led to a lot of questions now. Were they the carriers of plague? Were they running away from plague? Is that why they moved so fast? But also, these cultures were one of the earliest ones to domesticate horses as fast-moving transport animals. And so this suggests that maybe they were the animal vectors of these flea-carrying pestis. However, this is still actually quite an open question. This is quite new, and it will only be further sort of improved upon by contextualizing these ancient DNA results with the archaeological information. However, we are not just restricted to analyzing the genomes of single microbes, so just single species like Yersinia pestis. In modern biology, there's now this really hot trend or hot topic called studying the microbiota. So this refers to all of the microbial organisms living in a niche or an environment. This is much like the savanna in Africa, for example. All of the animals there have to live together in a harmonious-like way. Same thing goes to microbes. And all the genetic material of this is called the microbiome, which some of you may have heard of. And the human microbiota is a very hot topic at the moment in biological research because firstly, there is an estimated same number of microbial cells on your body to that of the body themselves. So in every surface of your skin, in your hair, your eyes, your gut, your mouth, everywhere is covered in microbes. But these are also very, very important. They are so important they're actually considered to be an extra organ, just as important as your heart or your liver. And the reason why this is because these microbes or microbiota have been shown to play critical roles in both the functioning of our bodies as well as health and disease. The famous example is your gut. So it's actually the microbes living in there that does the majority of the digestion of your food for you. This is a symbiotic relationship. You give it the food, which it can then basically get the energy from, but the microbes then break down these complex molecules in the food to the really small elements and compounds which your body can then absorb. But equally, when this ecosystem breaks down, like in the savannah, if you kill off all the gazelle, the lions are going to go hungry, and males are going to die. The microbial ecosystem lives in this balance, and if this goes out of balance and breaks down, it results in disease. With a dysbiotic microbiota being thought to be a major cause of IBS, so irritable bowel syndrome, or even things like Crohn's disease. And so there's also research into this to try and understand how the microbiome basically works. And we can actually access ancient microbiomes in the past as well from a range of sources. For example, as sort of Selina mentioned, I think earlier, there's paleofeces or even soil, which we can use to analyze the gut microbiome. We can use dental calculus to study the oral microbiome. On skeletons themselves, sometimes you get sort of mineralized nodules that can represent, for example, lung microbiomes in some cases, and also maybe also skin microbiomes from specimens such as mummies, although that's not so studied yet, so well studied yet. But why should we analyze ancient microbiomes in the context of archaeology? So everything that we do to our bodies will affect the microbiome in some way or not. So for example, behaviors such as diet can modify the microbiome. So for example, if you look at the oral microbiome, and potentially this can be a method of inferring behavior, not just today, but in the past. So for example, smoking, that will kill off microbes that can't tolerate the random gunk in your cigarettes. And also if you have a highly sugary diet, you might see an increase in sugar-loving microbes. And by comparing this through time, this would allow us to actually can we see how the microbiome is changing in response to the human culture or behavior, and conversely flip it back to actually infer what the human culture behavior was. And there's a few studies that already have tried to do this. So one major problem in modern research is that most of it has been formed in industrialized societies, which is mainly in the US. And it's known from the few modern anthropological studies that we have industrialized societies, for example, a lot of us, have highly reduced diversity of taxa, which has big implications for health because this can often result in disease. And in contrast, when you look at ancient individuals, for example, this is from paleofeces from Mexico and Iran and Germany, Austria, or somewhere in Europe, that these ancient individuals, even from the same regions as, for example, Europe, look much more similar to places from non-industrialized or less industrialized societies from around the world. So you see all the pink here in this top left box, and then a lot of these bacteria then disappear when you look in industrialized societies like Spain or the USA. And conversely, also industrialized societies have other bacteria compared to non-industrialized societies which don't have these bacteria, suggesting that obviously the microbiome is quite adaptive to whatever we are doing in our different contexts, because this is not consistent across geography, for example. And so by looking at microbiomes, we can look at not just the evolution of single taxa, but whole ecosystems of microbiota. And so now I'll go back to some examples of ancient microbiomes and what we can use with them. So again, back to bears. One well-known issue in biological sciences is the use of mass administration of antibiotics, which result in dangerous multi-resistant strains. And the overuse of these causes a range of problems, not just for humans in hospitals, because then we have no antibiotics to fight infections, but also in domestic livestock, where antibiotics were given to these animals to increase product yield by making sure the animals don't die. But then we don't know how the spillover from this affects the wider ecosystem. So what about learning how these wild animals which eat free-ranging domesticated get affected? And so in the 1950s, Sweden implemented one of these mass antibiotic administration campaigns to basically try and reduce the loss of livestock from infectious disease. But then they realized in the 1990s that this was not really going very well because of the rise of the multi-resistant strains. And so instead stopped that campaign as they became more and more aware of the wider effects. And then last year, in a preprint, Brile et al. Looked at the oral microbiome and dental calculus of Swedish brown bears over the last 200 years. And what they found was that genes related to antibiotic resistance in these microbes and from dental calculus started increasing in the 1950s throughout the peak of this campaign of administering antibiotics to wild animals, also humans. And then worryingly, even though the campaign stopped in the 1990s, it took at least 10 years to start seeing the amount of these antimicrobial resistance genes to start dropping down, to decrease. But furthermore, and almost even more worryingly, the diversity of, so the different types of these antimicrobial resistance genes increase and are still increasing even since the 2000s, suggesting that the microbes are becoming more and more resistant to many, many more different types of antibiotics that we may be using or want to use in the future, and also means that it makes it much more difficult to tackle this in wild animals. So again, that was more of like an ecosystem thing, but analyzing stuff like calculus is also relevant not just for studying the microbes, but we can also use to identify other bits of DNA in there. So the techniques we use to study the microbiome is called metagenomics, which, like I said, picks up all the DNA of all the things that might be in the sample. And so when you're eating, everyone gets food stuck in their mouth, as I'm sure everyone here experiences sometimes. And when this gets trapped for long enough and dental plaque forms, if this is left for a long time and mineralizes from dental calculus, this food DNA also gets trapped in the calculus alongside the microbes which make up the main bulk of the calculus biomass. And this has already been known to happen since the 1970s. And so there's a lot of archaeobotanists who love dental calculus, because when they demineralize the calculus, they can see lots of things like starches and phytoliths and seeds and things preserved in there, so they can sort of identify what may have been eaten by the individual. However, the problem with these sort of macro remains is that they're not particularly diagnostic to specific species. So it's hard to say they specifically ate this particular strain of whatever. And in one of the first ancient microbiome papers on studying ancient dental calculus of medieval women from Germany, Tina Warner, so you'll be hearing her on Friday, I think, in the keynote. In addition to studying different microbiota, they also found DNA sequences from different eukaryotic species, and these are basically what we eat. And so even though the DNA molecules are very, very, very low levels, they had sufficient information to potentially identify the taxonomic origin of the different foodstuffs. So for example, they found some DNA to sheep, some to cabbage, some to wheat, and also some to pigs. And now suggesting that the individuals potentially would have ingested these tissues at the time, likely affecting diet, and this sort of matches the historical record. And so this is still quite a difficult area of research because the DNA is so low, and that's because the microbiome is so rich and makes up the bulk of the DNA that is in dental-cochlear samples. But it shows that there are a variety of things you can learn by analyzing DNA from things other than just bone. And by looking at DNA, either from the human or the animals or the microbes, either the ones that kill you or don't kill you, but also live in harmony with you, we can really infer a lot about human behavior and culture, not just the biology of the human individuals themselves.
Yes, thank you so much, James, for this excursus into microbial biology. And to finish off this lecture, we're now going to look at a study that really combined archaeology, archaeogenetics, and linguistics. And to do that, we will go back to Vanuatu, which Annika introduced us to earlier. So you will recognize, hopefully, this graphic where they could show from the linguistic variation and the linguistic tree how Austronesian languages were spread across near and later also remote Oceania. And we'll particularly in this study, it's particularly about Vanuatu, which is an archipelago right in the border of near Oceania. And in this study, they actually they were the first to produce ancient genomic data from human remains from Vanuatu, and actually from this Lapita culture that Annika also mentioned, which have these specific patterns on their pottery. And these individuals were around 2,700 years old, and they could show in the study that these individuals were strongly Taiwanese related, so which confirmed that the Austronesian languages and were not just transmitted through cultural transmission, but also that actual people moved from somewhere near Taiwan into near Oceania. However, in this study, they also produced genomic data for present-day Ni Vanuatu, which is what the people who live in Vanuatu call themselves. And in these data from today, there was also a very large Papuan genetic component found in these people, in their genetic ancestry. And actually, when looking at some also archaeological samples again, this Papuan component was already present from around 2,500 BP. So when you look at this, again, another red mixture plot where the blue component here marks the Taiwanese Austronesian related component, and the green is the Papuan New Guinea related component. You can see that in the first couple rows, these are the Lapita associate individuals, the oldest ones, which show just a Taiwanese related component. Whereas from around 2,500, there's this influx of Papuan related ancestry, which slowly increases over time. And then in the modern individuals, which are at the bottom of this graph, you can see that they have both Austronesian and Papuan ancestry, but the majority is really Papuan related. However, what's interesting here is that the languages on Vanuatu are still mostly Austronesian related. It's actually, there's over 120 languages spoken just in this archipelago. But a connection to New Guinea and Papuans was already suggested from linguistic and also cultural features, since there are some connections between these two regions. And these are, for example, Papuan reminiscent linguistic features, such as the quinary numeral system, rounded labial phonemes, the dual exclusion of P and C phonemes, and serial verb construction. Whereas on the ethnographic side, there are shared cultural practices, such as large nasal piercing ornaments, penis sheaths, head binding, and also the rearing of full circle tusker pigs. So there was already an evidence in the linguistic and also in the cultural sciences that there were connections between these regions, which cannot be found outside of these regions. So here they could really, with the genetics, provide another line of evidence that supported this archaeological and linguistic so-called two-wave model that supposes that an initial unadmixed Proto-Austronesian speaking population arrived, probably with the Lapita culture that developed then, and that was followed closely by separate Papuan speaking and Papuan related genetic ancestry that made their way into the region. And I think this is really a really great example which combines all these different lines of evidence, and yes, I think is also a product of collaboration between the different departments at our institute. So just to wrap up the lecture, I'll just quickly go over what we talked about here in this introduction to archaeogenetics. So we started out talking about the history of archaeogenetics as a field, and how in the early days it was really just exploring and figuring out what actually works, until the development of next generation sequencing in 2005, and how the field has really been taking off since then. We introduced you to many different sources of ancient DNA, both directly from human skeletons, such as bone or teeth, and also hair, but also shells, calculus, paleofeces, sediment. With technological improvement, there's many new sources of ancient DNA being discovered every year. And then we went through what makes ancient DNA, and that's that it's damaged and fragmented and only around in very low abundance. And usually also samples are contaminated. And we addressed also how we try to avoid contamination and increase DNA yield in the lab. And then, of course, we talked about what we can actually study with archaeogenetics, which are, of course, as our focus is in the summer school, which is human history, and which includes the relationship with our ancestors, but also the migration patterns and interactions between different groups of humans in different time periods. Of course, animals can equally be part of ancient DNA studies, and sometimes it's really interesting to study the impact that human activity has on animal populations. And then James introduced you to the many ways how microbes are interesting to human history studies, and that we can also study the evolution of microbes themselves. But of course, this is only an overview, and you don't have to worry that you're not going to be tested on all this. We just wanted to show you all the different ways that archaeogenetics can provide information, especially in conjunction with archaeology and linguistics and cultural evolution as well.